Hello everyone and welcome to this class. In this class, we are going to study about the classification of protein toxins, right? So, uh, now protein toxin is very important, okay? So, as you know about antigens, right? So, antigens enter into our body, then our immune response work against it, and you must have read about the three kind of antigen which are protein based, then we have lipid based antigens, then we also have sugar or saccharide based antigens, okay. But in case of toxins, most of the toxins which are found in environment are proteinaceous in nature. That is why we are going to study about protein toxins, okay. So first of all, let us start with how these toxins are classified. So, first basis on which the toxins are classified are on the basis of source, okay, and then second is on the basis of structure, then third is on the basis of mode of action. Now, these three are very important classification bases to classify our protein toxins. So, first of all, we will start with on the basis of source, okay. So, let us say we have two bacterium. This first right here is gram bacterium, and the second right here is gram negative, okay. So in gram negative I'm going to make this orange layer right here. Let us say this is the lipid layer which contains here protein A, and this is LPS, lipopolysaccharide layer, which is found in gram negative bacteria, but this is not found in gram positive, okay. So in case of gram positive, what happens is it will form the toxin in the cell and continuously releases this toxin, continuously releases the toxins, right? So these toxins, which is continuously release or excrete, let us say excrete these toxins. Okay, so these toxins are basically exotoxin. Okay, excrete exo. Just remember it from this. Okay, so we have the example of these exotoxins as diphtheria toxin, then we have cholera toxin, and we also have tetanus toxin. Okay, different bacterium releases these toxins. Then, if we talk about endotoxin, then the case of endotoxin is a little special, as I have written here that lipid A of lipopolysaccharide. Okay, so basically this portion right here can act as endotoxin. Now, why am I saying that this is endotoxin? And I'm saying this because this endotoxin will be present inside the cell. Okay, it will be present inside the cell. It will not be exposed out of the cell, but it will be exposed when this gram negative bacteria right here will die. And after the death of this gram negative bacterium, the lipid A portion of the lipopolysaccharide will get released into environment, or you can say to host, and it will act as toxin. Okay, toxin are basically toxic substance which are not good for host, and we will move to the second basis of classification, which is on the basis of structure. Now on the basis of structure, these can be divided into three groups once again, one, two, and three. So first will be AB toxin. Okay, so assume that the this toxin as a whole occurs like this. Okay, this toxin or this protein toxin will have two components. We can draw it like anything, right? A B like this. Okay, so what happens is, let us say this is my bacterium. Okay, so what happens is the B part of the toxin will bind to the receptor which are present on the host cell. Okay, so here this B part will bind to the receptor, and it will create a pore on the cell wall or plasma membrane of the host, and through that pore the A part of the toxin will enter inside the cell and will cause whatever it is going to cause, like cytotoxicity or something else, like some protease activity, some enzymatic activities, inhibitory activity, anything can be there. Okay, so that is the property of A, while B, the property of B will be binding. Okay, so this is how AB toxins work, and the example of AB toxins are these two here in front of you, which is cholera toxin and diphtheria toxin. Okay, then you have membrane disrupting toxins. So their main target will be plasma membrane. Okay, they basically go into the plasma membrane and they show some activities which can cause the disruption of that plasma membrane, and by disrupting the plasma membrane, the cell will die or burst or whatever can happen. Okay, then the third category on the basis of structure is super antigens. Now you must have read about super antigen as you start studying about this from the 12th standard. Okay, so super antigens are the category of antigens which are basically, I'm categorizing it here as a toxin because it does act as a toxin because they stimulate a huge number of T cells. Okay, and they stimulate these T cells non-specifically, non-specifically. Okay, let us say this is my, I'm really sorry. Let us say this is my super antigen. Okay, so that this T cell will specifically bind to any antigen. Okay, so this interaction is very very specific. If let us say we have T cell against X antigen, then only X antigen should be here, then only it will bind to the T antigen. But in case of super antigen, it will bind to the T antigen of X, Y, Z, A, B, C, and many. Okay, so what happens is after the binding, or you can say non-specific binding of super antigen with T cell, what happens is T cell will of course release cytotoxins, and since a large amount of T cells are stimulated, so there is going to be a large amount of cytotoxin in your host environment, host or body, right? And this can cause many changes into our body. Okay, like inflammation. Then maybe NK cells might get activated in a large amount. Some other activities can be observed here. Okay, so this was on the basis of structure. Now we are going to move on the basis of mode of action. Okay, now if you talk about mode of action, then first will be your neurotoxin, second will be your enterotoxin, and third will be your cytotoxins. Okay, so in case of neurotoxins here, so the target is neurons, and in case of enterotoxins, it is related to your digestive tract, and third will be cytotoxic, though, so it will go ahead and kill the cell which is affected by that toxin. Okay, so that is how it moves. So let us start with the examples first. Okay, we have botulinum toxin and tetanus toxin. Okay, in these both toxins are neurotoxins. Okay, as you must have heard about tetanus toxin, that it goes and binds to neuron neurons. Okay, it stops the inhibitory action of the inhibitors which are released by the neurons, thereby causing a paralysis, which is, I think it was called septic paralysis. Okay, then we have enterotoxin, for which we have cholera toxin. So when we talk about cholera toxin, so you must have heard about the disease of cholera, which is basically a dysentery, right? So in case of cholera toxin, what happens is, now this cholera, let us say this is my cholera toxin. Okay. Now this cholera toxin will go inside the cell and it will show the ADP ribosylation of a G protein. So this G protein, it has regulatory function, and this G protein is found in the intestinal epithelial cells. Okay, epithelial cells. Now, due to disturbance to this G protein, what will happen is that there will be no regulation, and this everything which is basically your water and electrolytes, okay, they will move into your digestive tract and get flushed out of your body, thereby causing the problem which is diarrhea or cholera. So let's move ahead to the mode of action. Basically, this is pathway followed by toxin. Okay. So first, what happens is there is always binding and entry of toxins into the cell. Okay. As I have told you, just like the function of AB toxin, B will bind and A will enter. This will happen only in case of AB toxin, but in other cases, like if I say, let us say I have this XYZ, okay, so this will bind to receptors and will wholesomely enter into the cell. There can be other things which can happen while these toxins are getting bound to our cell, okay. So after getting bound and entering into the cell, then there is intracellular targeting, okay. So within the cell, our toxin will show two activities, okay. Either it will disrupt the host cell membrane or it will show enzymatic activity, in which it will show ADP ribosylation or protease activity, okay. In protease activity, it will destroy or denature the protein, basically targets the protein, and in ADP ribosylation, so it ribosylate your, what was that, your proteins or some other areas for in which ADP ribosylation can occur. Okay, as I have told you here in the example of cholera toxin. Okay, when ADP ribosylation. Oh yeah, I am sorry. Here cyclic AMP level increases. After the increase of cyclic AMP, then only the H2 and electrolytes will get released. Okay, so that was how it happens. And similarly, you can also quote some other examples of ADP ribosylation, just as if I take the example of diphtheria toxin. Okay, so in the diphtheria toxin will go and bind to elongation factor 2, thus it will disrupt the translation process. Okay, and in case of disruption of host cell membrane, hemolysin hemolysin can be hemolysin can be said as a major disruptor of cell membrane. Now third, we will move to the cellular effects. This is also the next step for the, I'm really sorry, this is also the next step through which toxins affect our body. So the first point, which is 3.1, here will be the protein synthesis, and 3.2 will be our cytoskeletal disruption. Okay, so cytoskeletal, let us start with the cytoskeletal. Cytoskeletal is a very important part of our cell, whose function is to give shape to the cell. It also provide, you can say, rigidity. That's why there is shape. Then movement of organelles is also a function of cytoskeleton. If our cytoskeletal get disrupted, then you know the function of cell will get disrupted and overall it will affect the host. Similarly, if the protein synthesis is inhibited or somehow interrupted, or you can say in some cases there can be a case when protein is not manufactured or it can become non-functional when toxin enters into our body. As I have told you here, in case of diphtheria toxin, if elongation factor 2 will be affected, then no elongation will occur, and at last the inhibition of protein synthesis is observed, which comes under the cellular effect for toxins. Then fourth will be the induction of immune response. As I have told you, in case of super antigens, okay, what they will do is they will stimulate T cells in high amount. Then they will release cytokines again in high amounts, and this thing will go on and go on, and ultimately very much functioning of our body or host cell will get affected. Then we have systemic effect. Now systemic basically it relates to blood, okay, which is fluid connective tissue of our body. But here, uh, not just blood but organelles, organs, organs of our body can also get affected. Now, since toxin, if enters into our body, then it tends to move from one place to another, or you can say from one cell to another. Okay, so from moving or following any path from one cell to another, of course, there are chances that it can affect various organs like liver. We have liver, then we can also have lungs. In worst cases, brain, spinal cord. Okay, so these can also be observed in uh, protein toxins case. Now, this was the mechanism basically. So that's it for this class. Thank you very much and see you soon. One, two, three, check, check, check. Illuminati लगा दो, चलो